Hello friends, welcome to BMH Learning. This lecture is about protein targeting to mitochondria. Some key points before we start. Protein transport to mitochondria requires the input of energy, and that's mainly provided by ATP hydrolysis. The mitochondria has double membrane system, the outer membrane and the inner membrane. Both the membranes of the mitochondria has pores or translocations through which the protein translocation takes place. So, for the protein to reach mitochondrial matrix, first it has to go through the pore which is present in the outer membrane, and then it passes the channel which is present in the inner membrane. Pores in the outer membrane are designated as TOM, that is, for translocon of the outer membrane. Similarly, translocating channels of the inner membrane are referred as TIM, for translocons of the inner membrane. Protein import occurs at points where the outer and the inner membranes are in close contact. Now let's discuss the mechanism. Suppose we have this precursor protein which is to be delivered into the mitochondrial matrix. The N-terminus of this protein has matrix targeting sequences which direct the protein to mitochondrial matrix. These matrix sequences are amphipathic because they contain both hydrophilic and hydrophobic amino acids. For the easy uptake of this protein, it is usually kept in unfolded state. The unfolding of this protein is maintained by cytosolic chaperone proteins known as HSC70. This process of unfolding of the protein by HSC70 requires energy, which is supplied by ATP hydrolysis. Now, import of this unfolded protein precursor is initiated by the binding of matrix targeting sequence to an import receptor present in the outer membrane. This import receptor is called as TOM20/22. The import receptor then subsequently transfers the precursor protein to an import channel in the outer membrane. Now this translocon is named as TOM40. For the protein to get entry into the mitochondrial matrix, we bring another translocon, and that's found in inner membrane. We call this channel as TIM23/17. So the protein starts to translocate into the mitochondrial matrix through these channels. Now there is a question: who is triggering the intake of the protein? So there is a matrix chaperone also named as HSC70. This protein interacts with the transmembrane protein known as TIM44. Now, the interaction between these two stimulates ATP hydrolysis by HSC70. Thus, the two proteins act together to power translocation of the proteins into the matrix. As soon as the N-terminal matrix targeting sequence slips into matrix, it is recognized by a matrix protease, which removes the matrix sequence. But why? Because the protein no longer needs it, as the protein has already got entry into the matrix. Finally, the HSC70 is released and the protein gets properly folded, and this is our active protein. This was all. Thanks for watching.